So I'm going to start recording now. And it is recording, yeah, so be careful what you tell, because I know some of you have results that have not been published yet, so you might like to be careful there. So let me go back to the slides. We have a full agenda, as you might have seen in your invite. For those of you which joined the very first time, welcome. This is the ASCT table meeting, which came out of the CCF session at the joint NIH-HCA meeting, which happened recently and was supposed to happen in DC, but then it all was virtual. At that event, there were many very important and interesting breakout sessions, and one of them was on the Common Coordinate Framework, the so-called CCF. And in that session, we identified the need by many different consortia, which you see here listed as column headers, in identifying anatomical structures, cell types, and biomarkers for 30+ different organs. And so in this table, you see different organs cross-tabulated with the different consortia efforts, and you also get to see how many organs and which organs they currently have as focus organs. I would also like to point out that HCA, in the meantime, has created a website for biological networks. Sarah Teichmann presented this at the HubMap annual meeting. She was not able to join us all here today, otherwise she would have presented this work. But as you see, it's a consortium of experts from HCA, from HubMap, like for instance, Mike Schneider is here listed under GUT. And I think also Xin Lin joined the HORT in the meantime. So I think it's a very inter-consortium effort to try to understand and get under control all that new data that now becomes available. And so if you have not heard about this, you might like to click on this link. And again, the link to these slides is in the group chat. At the second ASCT table meeting, we discussed tables for the kidney, liver, and for intestine. We also renamed those tables into ASCT+B tables because the biomarkers are actually quite critically important for understanding what kind of biomarkers are used to identify what cell types. We also now have a draft editorial process to author and review and ultimately approve these ASCT+B tables. So again, this is the link to a document in process. So all comments and questions are very welcome here. We also had a number of ontology experts joining us, and more are joining today, to help us identify efficient ways to link these ASCT tables to organ-specific subsets of existing ontologies. And here we talk especially about Uberon, but also cell ontology. And going forward, we of course want to also link to UniProt for proteins and Go for genes and other ontologies that describe these biomarkers. In today's meeting, we would like to introduce better ways to organize this work, because we want this to be a very productive and rewarding exercise. And so we now have a roll call sheet, and we can go over to this call sheet. And I see many of you busy, so thank you for doing this. Please do put an X whenever you attended one of those meetings. This helps us understand who has heard what kind of information. But also this way we know what experts we truly have right now. And as you will see, we have calls for experts where we need specific expertise, for instance, for a lung ASCT table, which we would like to create. And so please do go over and add access. And thank you so much for helping us understand who has been in which meetings. Back to the slides. We also now have a Slack channel. So several of you, and I guess that's the channel, several of you have joined that channel. I and Ellen invited all of you, but oftentimes the invite actually ends up in your spam folder. So if you could please go over to your spam and try to understand if you received an invitation from me or Ellen or Slack in this case, and then join this channel if you want to discuss with other experts issues surrounding those ASCT tables. And as you see, we have many experts joining, And we also have organ-specific channels now for the heart, the kidney, the skin, the spleen. And we have some first adopters which will be moderating and helping organize discussions surrounding those organs. We also have created Excel file for expertise surrounding these ASCT tables. And Ellen, if you could also put that particular link in the group chat, then I can also get to it. And in that particular Excel file, we tried to understand who has what expertise. Some of us have are engineers like myself, and we have information technology and information science and visualization expertise. Many of you have organ-specific expertise. Some of you are anatomists and pathologists, and potentially anatomists and pathologists might see this world from different angles. And so it's actually very important for us to understand who created a certain organ-specific table, because what we want to accomplish in these calls is that we all speak about the same language. Maybe we can't get to perfectly the same language because different organs will inspire different ways of seeing this world, and they have different functionality and different functions, and therefore they might need a different type of organization of anatomical structures and cell types and biomarkers. But as much as possible, we would like to agree on one language that is the same across these different organs. This is really valuable for HCA, HubMap, but also for many, many other efforts. And so again, it's very important for us to understand who is coming from what angle. And again, watch out for the Zoom group chat so that we get your information in there. We tried to pre-populate and pre-fill, but we don't yet know all of you 100 so well. And then for today, we have presentations and discussions of some new and some more advanced ASCT+B tables, about 20 minutes. And we will start with the heart and then go over to small and large intestine. And then kidney vasculature. Griffin was actually not available. He's meeting with his dean right now, so that's a good excuse. But I will cover some of this in the Making ASCT-B tables machine readable. And after that, we will have David Osumi-Sutherland present for 10 minutes on a comparison of the current tables we have and Überon terms that exist in the Überon ontology, which is a very, very comprehensive ontology, and we wanted to understand what the overlap is there. We will also have, last but not least, a brief demo of anatomical structure pathonomies as they are used in the CCF exploration user interface that was designed for HubMap. And Bruce Hare will give this demo about 5 minutes. So let's go over to well, the roll call sheet. So if you haven't made your access yet, please do. And there is the link also here. So if you just have the link to this slideshow, you just need to click on these links. But also the ASCT-B table authors, reviewers, and other experts, please do help us understand where you are geospatially, but also topically, and what your role is in these calls and in this effort to create these tables. And we already looked at the Slack channel, so let's go directly into the presentation and discussion of new ASCT tables. And Xin Lin, sorry for misspelling your name. Xin, if you could please, together with Mark, or maybe it's Mark who present, because Xin Lin is also back into in the hospital, and so he might be operating. So Mark or Xin, if you could please present on the heart.
Great, yeah, so I'm here. This is Mark Halishka. I don't think Xin is on the call because I think, as you said, he's back clinically working today, but Xin is a cardiologist and I am a cardiovascular pathologist, so it's been nice to have this interaction. We actually share the same mentor for our PhDs, which was also somewhat funny, but at different times. And we tackled the heart piece of this. So the heart is an interesting organ because structurally there's many different parts of it, which is fairly unique outside of perhaps the brain. You have valves, you have myocardium in the ventricles, but which are different. You have atria, you have blood vessels, particularly coronary vessels. There's a lot of different structures that were there. And so what we basically did was throw in all those major structures, which was based on the clinical knowledge we had, as well as we'd gone to Uber on and made sure there was nothing we were missing from that experience. So then we listed all the different cell types that we knew to be found in these different structures. We used the expression cell type data based on this very recent publication of Sarah Teichmann's laboratory, where they did just a fantastic job by single cell characterizing different cell types. So you can see it here, that's exactly correct. And that's the link to it's on bioRxiv currently. Now, one of the things that we debated, Shin and myself, was how deep a dive do we want to go? There are cells that have been described by some people called telocytes or interstitial-like cells of Cajal, which didn't show up in that single cell paper and really aren't well described. In general, do we want to include something like that, which isn't well known, and we don't even know what the markers are for those cells necessarily? It's more of an electron microscopy diagnosis. Whether there's structures that we wanted to include, for example, the moderator band, which is a piece of muscle that runs across the right ventricle, or the AV node, which we think is important, but we don't yet have samples from there to help us understand the cell types that are present. So those are some of the gaps in our knowledge, and a discussion is really how deep we want to go into some of these areas. And so the plan going forward is to really work with single cell RNA sequencing groups to capture some of these rare regions of the heart if they're interested, if we're able to get those samples, and better correlate unique cell types with proposed cell information from the single cell RNA-seq data set. So we think overall the heart's in a really good position, that we really on top of, we know the parts, we know the cells, and now with the single cell data, we know some good markers of these cell types. And the only question is, how much of lumpers or splitters do we want to be of dividing this stuff up? And I'll stop there and take any questions.
I very much like that you added the link here. So again, if you go to the tiny URL and then to the heart tab, you can get to this table and read it in large. And again, this is the link to the bioRxiv paper. I also really appreciate that you started on the vasculature, because that's very relevant for using the vasculature as a way to map every single cell in the human body. And I will tell a little bit more about that effort later. Thank you, Mark. But I think there are others which wanted to talk.
I'm definitely admitting not to be an expert here by any means, but in the conversations that I hear, sometimes I hear conversations around the evidence for a cell type. So not necessarily like, will you include it or not, but if you do include it, would you give it like a prior or an estimate, or like what kind of evidence supports this, especially in these times where we're still growing the conversation. So it might be a perspective to think about. Yeah,
that's fair. And I think that's just a fundamental question we're all struggling with, is where do you draw the lines between cell types based on expression only? Endothelial cells is a great example. It was an endothelial cell of an artery versus a vein. Is that two different cells? Is that one cell type, endothelial cell, that has different expression patterns because of the pressures of the blood passing by, as an example, or capillaries? Again, these are really fundamental questions that I think all of us have to think about, and then we can respond in these sorts of tables when we have some general ideas from the larger group.
Are there other questions, comments?
This is Mark Musin. Is there a downside to including putative cell types just so that we don't lose track of this kind of information? I'm worried that there's a possibility that some of these cell types, which may not be confirmed histochemically, might still be relevant in the future. And if we lose them now, we may never get them back.
Yeah, that's a good point. The one that, like I said before, the telocytes is something that's very rarely studied, that we could probably put in, but I don't know enough about what specific markers we would use to pull that out. But I think that's a fair point. We don't want to lose anything up front.
Very
Okay.
good.
That's
Thank you
great.
all. And Mark, thanks so much. And actually having a quiet moment in these things to reflect and think about things, this is very unusual, but I think it's actually very helpful. Gives us all a tiny micro break in these Zoom to Zoom meetings. So I will now go over to the small and large intestine. And I wonder, Aaron, who from your team will present?
Sure. Hi, everyone. I'm Aaron. I'm a postdoc at Stanford, and I've been working with a group of individuals, Ying and John, and also Mark Ahrens, and a few others, Stephanie Nevins, and also Winston Becker from Stanford, to put this table together. So I'm happy to show what we have so far. Yeah, there we go. So this is the top portion of what we have at the moment. But actually, lower down we have an updated version of our table. So first what we did, because the colon and the small intestine are so repetitive, first what we did was we filled in just the top section of the duodenum superior, and that's part of the small bowel. And what we found was that there's mucosa, submucosa, muscularis propria or muscularis externa, depending on what region of the small bowel or colon that you're in, and also serosa. And so basically we just repeated those throughout the entire length of the small bowel and colon. Now, of course, these tissues are a little bit different. And so in realizing this, we had to make a guess as to, well, a guess, we had to make a plan basically as to how we were going to build this table. So we first developed our small bowel duodenum superior, and then just copied it throughout the rest of the, throughout the rest of the tube organ. And so everything else is just copies of basically the same portion. So of course there are some differences between the small bowel and the large bowel, and that's not only in the vasculature, which was shown at the top, but also, yeah, so right down there, that's the original table that we had developed with the initial plan for everything. So there you can see we've got mucosa, submucosa, muscularis externa, and serosa, or muscularis propria across everything. And then that top portion was basically copied for the rest of these. But I wanted to hold that there as like a structural kind of plan. This is how we went about this. But then we've also discussed, okay, so the Paneth cells in the small bowel, they become Paneth-like in the large bowel. So we need to make those kinds of changes. Additionally, the cell markers, the actual genetic markers, are going to be a little bit different. So we also included a column there for the gene markers. So all of that right there is only for the small bowel, but I have still copied it over for the large bowel, but that's probably going to change. And so we are doing some single cell analysis, and through that single cell analysis, we'll be able to get better ideas as to which markers actually help define these different cell types. And that'll be, positive and negative gene markers. So, is it positively expressed in a cell type or negatively? And we also have discussed, rare cell subtype issues, but we figured that at least initially for the first pass of developing this table, we wanted to get things that we know are there, that are canonically known to be there. And then maybe in later versions of this, because I believe this to be a living document, this will have more changes in it and more cell subtypes. Additionally, if you scroll up to the top, you'll see that we do have some ideas to what the vasculature is like in these tissues. And of course, Mark Ahrens or Ying, if you guys are on, you guys can discuss this a little bit more, but it's pretty widespread. And so I haven't quite figured out yet exactly how to add this, but there of course is vasculature there. But it's just a matter of figuring out how to put it into these tables.
Thank you so very, very much, Aaron. And I think your team gets definitely 3 stars for having the longest organ, but also the most appropriate, most longest ASCT table coming with it. Thank you so very much.
A lot of it is just, repeats of the same thing. So, it may seem long, but it's a lot of the same stuff over and over again.
But I think your team also was among the first to list not just marker genes, but also proteins. And I also really like to see positive and negatively expressed. And then as we have tried for the kidney, actually Griffin Weber did this, who is not going to be able to join today. You also try to capture the nervous system and the lymph system and the vasculature system. And that's, I think, very, very important information as well. So thank you so much for going for the full challenge.
Yeah,
Let's
a lot of
see,
thanks goes to Ying and Mark Ahrens for that. So thank you guys.
Thank you all. Yeah, let's see who else has comments, questions.
On the general approach of these repeated.
Can you speak up, please?
Sure. So I'm curious about the approach on these repeated segments. That's just the reality of what's presented to us, but in terms of structuring tables and ontologies, is this the way to do it? I don't know the answer.
That's a good question. Yeah, I'm not sure either, and we definitely struggled with it, but you know, we weren't sure, and we decided to go with, you know, the simplest route, which may not have actually looked all that simple. But yeah, it's a good question, and I'm not 100% sure the way to do it.
And I think, isn't it going to in part depend on what you find at the biomolecular level too, right? Because if there are differences at that level, then that will need to be captured, and we won't know that right now.
Yeah, I think that's one of the reasons why I think I pushed for having it be just this full long document, because we may end up finding those differences that make these regions more distinct, which we suspect that they are, you know, pretty distinct to some extent. And so, yeah, having it all there just made sense. Yeah.
And Aaron, I know you have 8 sampling sites, so soon we will know if it's the same or very different across those sites. Similarly, for the kidney, we also now have samples extracted from top renal pyramids versus those in the lower part of the kidney. And so currently we don't know if there's a gradient going on. And based on this, we might also have to structure it differently. And I'm so glad we have some ontology experts here on the call so that they also get to understand what interesting research challenges exist in all of this, also for ontologists. Mm-hmm.
Certainly, we know that there are some proteomic and lipidomic and metabolomic differences between the small bowel and large bowel, which makes sense. They have different, you know, functions in terms of the way that they metabolize food. And also, you know, I would expect that there would be differences between the left and the right colon based on their propensities for different types of cancer. I mean, I would just expect that there would be differences. So they may be minute, but they're probably important.
But at the moment, the markers you've listed, you know, are there in all these segments.
In the small bowel, yes. We know that those are there. I don't know if Winston or Stephanie are on the call right now, but they've been working with the single cell data, and they were the ones that put the, and also I think John's on the call. John can probably speak to this about what the markers actually mean for the large and small bowel and the different cell types. John, can you speak to that about the differences between the regions and the cell types that we chose?
So yeah, we haven't gone into that specific of detailed analysis between the different regions. There's obviously differences, but I think we were holding off until we had the single cell sequencing data to integrate with the Codex imaging data for that.
Thank you.
Yeah, I'm wondering, like the earlier comment on capturing putative cell types, I wonder if we need to have a way of flagging putative markers when we have an expectation, but we're lacking proof at a certain point.
Yeah, Clive, yeah, I was going to add to that. I think the ileum is where vitamin B12 is absorbed, and the cells are probably all got the same markers except for a receptor for B12 and intrinsic factor or something like that you wouldn't normally see by some of the processes. So we can get really lost in the weeds and the details. We've encountered this with spleen, for example. Is this, in theory, with deep sequencing, we should be able to say, at some point, this is the T cell that recognizes CMV, but it still looks like a T cell normally. It's going to be really challenging to get down to that level, but in theory, possible at some point.
Yeah, these are good points.
Any other comments, questions? All right, so let's go back to the tables. And again, you should have a link to these slides, and everybody can add to these slides, which is a little bit scary on my end, because if somebody deletes them right now, that would be that would not be good. So be careful who you share these links with. But please do also add other experts that have contributed, Aaron. So I would love to have them in here. Going to the agenda. So as I mentioned before, we are very interested to make these machine readable, these tables, while keeping them human readable, which is a true challenge because machines like different inputs than humans. However, for the purpose of actually using these tables to then create OWL files, for instance, of autonomies and cell typologies that are then showing in the exploration user interface, which Bruce will demo, but also for then automatically labeling some of the 3D structures in the 3D object library we are compiling for HubMap. We just must have them machine readable. And this way we can also cross-check them against Uberon and Cell Ontology and Uniprot and Go, etc. So we have been trying to flatten, so gently make them machine readable in a way so that experts can still read those tables, but also now machines have a unique way of parsing that data. And so what you see here is the vasculature for the kidney, which Griffin Weber compiled. And then we started to flatten these tables so that you have basically one smallest anatomical structure. So one of the leaf nodes in your anatomical autonomy representing one row. And in some cases, however, you have OR combinations of marker genes. And if there is an OR combination of marker genes, then basically you have two rows in that particular line. And I think that's potentially the middle point where humans can still read it and machines can also read it. And this way we don't need to implement interfaces, which would take us a while, but we can start having these tables machine readable. So several on our team have started to flatten those tables, and those of you which have created tables, you will soon get to see a flattened version of your tables. And we hope that you can help us improve and correct as needed. But then these tables can now be read in a way so that you can.
Kati, are you trying to show something?
Oh,
because
yes.
Sorry.
Thank you, Serena. So yeah, I was assuming you see this. So here is the table for the vasculature for kidney in the top part, and in the lower part is the flattened table where you see the tree structure. So now an algorithm can actually just read this part of the table in. So it would just take those leftmost columns. It could also read that tree structure and then do a bimodal network of the anatomical structures and the cell types, which is another important view, I believe. You might also like to have a typology, so a tree structure for your cell types, for instance, grouping them by vasculature relevant cells, by immune cells, and by other cells. But you also ultimately will have another bimodal network, which is the cells or cell types, if you want to aggregate it up, or if you have to aggregate it up because you don't have detailed information, and the what I call cell calling cards, the marker genes or proteins or other biomarkers that are typically used to identify these cell types, including stains and other means to identify cell types. So you now can use algorithms to very easily extract these two trees, the autonomy and the typology of cells, but also to have their interlinkages and ultimately to have the interlinkage of cell types and the different other ontologies that exist for gene types or protein types, etc. And so I think in this way we can then connect it much better to the linked open data and use it also for some of the work where we are exploring using the vasculature for mapping the human body at the single cell level. So we need to go from the whole kidney level, in this case, from the organ level, extract the vasculature, go down to functional tissue units, unfold that again, and so go down to the scale where you actually see single cells. And even though we all might have rather different concrete pathways or vascular structures here in this case, but also lymph and nervous system structures. Ultimately, we will have probably the same subset of cells that is fed by those three systems and aligned with these three systems. And in order to get there, we really need also the vasculature in these ASCT tables. So I very much appreciate that we now have some of those tables which have the vasculature included. Griffin Weber would be the one who would have gotten more airtime for this, but given that he can't join today, I think we let him talk about that part of the work next time we meet. And by the way, if you haven't seen this yet, so the next two meetings are in this corner up here. So if you want to mark your calendars, please do. I will send out invitations after we know if there are any new additions to the meeting invite after this call. So next item on our agenda is David's presentation of ASCT plus B tables in Uberon. David, are you on the call?
There we go. Sorry, just unmuting.
And
yes.
Chris is also here, which is wonderful.
Oh, great.
Welcome,
Brilliant.
both of you.
Okay. So let me. Okay, it's the same slide deck, but I'll just share myself here. Just a minute. Oops. Okay, thank you, Cathy. So really encouraging work going on here with a pretty huge community contributing, and I'm really interested to see how we can kind of fit that together with all the work, the ontology work that's gone before, to try to put, which, you know, has put together a lot of this information in some form, and has been used, you know, a set of ontologies that have been used quite extensively in biology to annotate lots of data in bioinformatics. So the kind of core ontologies I think we're interested in here are is Uberon multi-species anatomy ontology and the cell ontology. Now, both of these are, so Uberon has very extensive curated system of cross-references to other ontologies, so it's kind of quite a nice point to map out into biomedical ontologies, for example, like SNOMED, NCI Thesaurus, and UMLS. It also has connections to lots of other anatomy ontologies and various artifacts, including Partonomies from the Allen Brain Atlas, which I haven't shown here, and links to the foundational model of anatomy. Apologies for the formatting here; it kind of went weird uploading to Google Slides. And also it's actually directly, extensively used. There are direct link part relation links between the cell ontology and Uberon, and there are direct, Uberon is used to define terms in the human phenotype ontology, which is used massively to define human phenotypes. In the disease ontology, Mondo, and in the gene ontology, both. So all of these use the cell ontology and Uberon. So it's kind of quite a nice integration hub in a lot of ways, as well as being kind of a store of knowledge. It's also used in lots of single cell annotation. So here's some examples of cases where it's being used in single cell atlas, HCA, DCP, and this BG database from SIB. And I've been working on using this nice integrated query point from RENCI in North Carolina, developed by Jim Balhoff. And this pulls together Uberon and the cell ontology and a bunch of other ontologies, and allows us, and it pulls it together in a way that kind of takes advantage of all the knowledge and collapses it down into quite a simple form that's very easy for us to query and integrate with the kind of things that you're building. So, oops, now how can I, sorry, there we go. So I've been taking the spreadsheets that have been developed so far. I've taken the spleen spreadsheet and the kidney spreadsheet, and I used the sort of flattened versions of those spreadsheets. Here you can see the kidney partonomy represented as an indented representation here, but the flattened version of that is very easy to script against. There are nice mappings to Uberon already. So the first thing I did was to try a validation. So we have a big graph of part relations between parts of the kidney. So I checked the kidney and spleen, and actually they validate very well, which is nice and encouraging. There's the vast majority turn out to be things we already have. We have a lot more detail in Uberon, but this query interface allows us to collapse that down. So the only thing I found a few cases, so having we don't have the ureter as part of the kidney. That's probably a modeling thing you want to look at and maybe question, because clearly parts of it are outside of the kidney, and that was under your graph there right now. Then we have some cases where there's I've been assuming all of the hierarchy represents a partonomy here, but sometimes Uberon interprets it as a classification. So we have major calyx as of the kidney as a type of kidney calyx, not as part of it. So there's a few kind of small, but those are kind of minor modeling issues. And then Uberon doesn't have renal tubule part of kidney, but I think that's because of Uberon's kind of multi-species brief. So in that case, I think, you know, for the human anatomy, that's trumped by what you're doing. So that was the first step in validating. And actually, I also validated the CCF AL file that you produced before this process. And actually, I would say I got less errors taking the new spreadsheets than I did taking the original AL file. So that's really encouraging as well. What we can also do, we can pull out visualizations of how it looks in Uberon. So here's an example of a visualization of the part relationships that I pulled out of Uberon. So we could use that to look for anything that's missing, for example, and to inspect how Uberon models. I can pull out cell types. So here's an example, cortex of the kidney. We have a term that's not in this list here, cortical collecting duct. And then underneath that, we have a couple of different cell types. So it'd be interesting once you start to map the cell types, and we could use that to generate lists of cell types that we already have to help your mapping. And we can try and validate and cross-check the part relationships we have in the cell ontology with what you're building. And we can easily pull out cross-references here. So that, I think, you know, we've got a bunch of proof of concept queries, and we can start to build pipelines that actually help cross-integrate these two things. And I think that's, you know, it can help make Uberon better, and hopefully it can help make the CCF effort better as well. Okay, so that's all I have to say. Is there any questions on that?
David, thank you so very, very much for all that work that you did. And thanks also for taking on these different types of files and making them work in your infrastructure. That's non-trivial. I think it's very good to see that there is some correspondence already, but it's also great to use Uberon to validate some of the initial tables. We have also been discussing to use these tables to seed future table creation efforts. Sometimes that's very intimidating. I think for the brain, there are about 1,000 Uberon terms just for the human brain. So potentially experts asked to organize those 1,000 terms could just say, well, I don't have time for that.
But I guess for the brain, you already have the stuff you already have all of the work that's had a lot of community input of the Allen Brain Atlas. So presumably you could use their autonomies as they.
And actually, Amy Bernarci wasn't able to attend today, but we have been going back and forth about the 141. So 141 parts that are also modeled in the new 3D Human Brain Atlas. And so there were some back and forth about some of the terminology. And yeah, I think it's wonderful that actually for the Allen Brain Atlas effort, we have the ASCT tables. Actually, we only have the anatomical structures table, not yet the walk over to cell types. I think that's BICCN working on it. But ultimately, we also have for those 141 structures, we have this 3D model just for one half of the brain. But apparently that one half was modeled in a way that you're supposed to mirror it to have the entire brain. So that's the state of the art in terms of organ model for the human brain right now. And it's wonderful that we have this. So let me open up for any questions that might arise.
Katie, this is Gloria. I do have one question. I guess I wasn't certain as we get started with the long table, ASCT table, whether you would like us to go to Uberon and work from there, or what I just heard was perhaps better to work independently and then rely on the validation that was just presented. But what is the recommendation? What do you think?
So I believe you're all experts. So what I'm going to do is to post into the group chat a table that basically has a listing of anatomical structures and their Uberon numbers. And it has it for as a summary. So you actually get to see in the summary tab of that table. And here, let me screen share. David, if you don't mind letting me show Uberon numbers, I think it's all in the spirit of
oops, sorry.
of really making Uberon shine here.
There we go.
Okay. Let me share this here. All right, and then I can make it larger so that you all can see it better. All right, so in this table you have a summary tab, and in that summary tab you get to see how many anatomical structures are involved in part of relationships. So this is the view of Uberon just in terms of part of. I know there are 30 or 50 other relationships that all of you might care about, but right now it's just part of relationships. And you get to see that we have many anatomical structures for the brain and many relationships and many paths from anatomical structures to other anatomical structures. But we also have those for the lung. So for the lung, we would have 134 anatomical structures, which seems manageable, Gloria. Maybe that's not too intimidating. And then we also have relationships. So you could go to the lung and you could look at all of these and you could say, oh yeah, that's, as an expert, I totally agree with that. Or you could say, maybe we can improve upon that. So again, I think it's now up to the experts to decide if they want to benefit from the existing knowledge that many other experts have contributed to Uberon, or if they want to just create their own view of the world and then compare it with Uberon. I think both have value.
Kati, I have a question. In your table on the vasculature, you had 0 for many of these things. I'm assuming that means there were no people who annotated vasculature in those organs, because otherwise there are no organs that have no vasculature.
Exactly. Yeah, that's correct. Yeah. So that's, I think, where we really need experts to help us with identifying these vasculature structures.
It's the skinniest structure and it's the one that connects everything.
Yeah. And again, if we would have looked for adjacent to or feeds or passes through, we might have identified more, but this is really part of
every functional unit of every organ that actually creates the identity of the organ contains vasculature. So it should be at least one.
And maybe there was, again, a different view of this world. So again, one of the main purposes of this call is to agree on a common language that we can use across the different organs that HubMap and HCA and many other efforts care about.
So is the issue here that you did a query in Uberon for vasculature of the adrenal gland and there was no terms instantiated for that? Or is this more about the previous work on building the tables?
This is trying to answer Gloria's question. Should I start with an existing list of Uberon terms and part of relationships? And she can use it. So that's up to her to decide to just go and use this. We have provided this now to the organ experts to make it easier for them. And also in some cases, it's easier for us at the end, because if they would use these terms, we don't have to manually try to understand if these two different terms refer to the same anatomical structure. And as you know, that it's not unified yet.
So, David, what do you do in your analysis if a term is ambiguous? Because you're mapping from the, are you doing named entity recognition to get
No. I mean, so far I've just taken, so for the mature ones, there's just a list of Uberon terms already. So all I'm doing is taking the Uberon terms and their partonymy and comparing to, because we've got that collapsed down, partonymy in with inference in that Sparkle endpoint, then I can just check. I can just validate each part relationship and see if it holds. So yeah, and that
stream of
yeah, that, and then I can take the terms and just rebuild a non-redundant partonomy from that as well, based on what's in Uberon.
Okay.
And pull in the cell types as well if we want. So.
Hi, I'm Kathy Reinhold from the Broad Institute. I'm working on HCA, and sort of more to the point, I'm working on a common data model to use across data at the Broad. So I'm probably looking too far into the future, but when will these terms become part of Uberon? And secondly, I use extensively ontology servers like OLS to research the terms. Do you recommend doing that as well? This list in Excel, for me, it's a little hard to use. So.
Yeah, I mean, the terms, all of the terms so far are in. I don't think there's very few missing anatomy terms. There's actually a lot more detail in a lot of cases in Uberon right now. So I think there's a lot of missing cell types. That's definitely not complete. There's in some places it's reasonably complete, you know, but apart from all the newly discovered ones.
Do you intend to keep cell types in Uberon though, or are you going to leave that to the cell ontology?
No, so they're cross-linked. So there are many. So the cell ontology imports terms from Uberon and makes part links to them, so that you can cross-query. So they're kept as separate ontologies, but one imports the other.
Now, I believe if you look at the cell ontology in OLS, it brings in all the parts of Uberon that are needed to kind of house all of those cells.
That's true.
So you should be able to browse the interlinked structure on OLS. One thing we could do is for this project is set up a special OLS instance that is a bit more customized for the work here. I know you've done a lot of work in that area, David.
Yeah. Well, it's other people in the group have been working on making that easier to launch. I know that there's one running at Monarch now, so it should be easy enough to give you a container if you've got a server, and we can just specify the setup. So if you actually want to run a local instance of Uberon with particular slices of
I have a
information,
question. I have a question for something like vasculature that goes across scales and across the body. Is there a way of extracting all the terms that have been already associated with vasculature of various organs and see what's missing, or how would we go about? I mean, see if there are sort of terra incognita in some places. It looks like in the kidney you have some, right, Kati?
Yeah. So we have now for the heart, for the kidney, and also for the colon, we have beginning tables for vasculature, but they are not extensive yet.
So, for instance, actually the endothelium is the largest organ, not the colon. But anyway, the endothelium goes from everywhere to everywhere. So you should be able to find that thread in Uberon somehow. And if it's interrupted somewhere, it means that information is missing. That's where we need to go and
we should be able to come up with some queries for that. I'll probably need a bit of Chris's help to understand all of the modeling, but it should be straightforward. It's not all going to be part relations, yeah.
Exactly. Yeah.
Because I remember when I was talking to Kati and Griffin and so forth, I found several mostly medical ontologies for radiologists, how to follow, because the vessels are basically major landmarks every time you try to take a biopsy or do surgery or whatever. So there are all these medical ontologies that go all the way to the very small vessels, not quite at the level of the cell, but they go across scales, and that should be easy to sort of somehow put back into the various organs. But I don't understand really how Uberon works, so I'm telling you the pathologist view. I can see them, I can follow them with various imaging, different scales. How do I?
Yeah, if you have the terms, if you have the IDs for terms in other ontologies, they may be. You may find that they're mapped in Uberon already, so we could query and pull those out and use that as the starting point as well.
Can I add? This is Mark Klishka again. that the named vessels tend to be exterior to the organs, that generally once you're into the organs, they get to smaller arteries, arterioles, they have more random courses, and they're not named as much. There are some that are, but generally not. So the stuff that people talk about, the radiologists, are really the larger vessels they can see, and those are the ones between the organs, not in the organs.
Mark, but that's the case with every other cell. No cells have, I mean, there's a cell type. So you get back to the level of the cell type. So it will be endothelial cells of a different type. It's the same thing with any other cell in the kidney. They don't have individual name, they have a cell type. So I don't think, exactly what I said, I think in the medical imaging and ontologies, you can go all the way to the cell level. And when you get to the cell level, then you need the microscope to say, this is an endothelial cell of this type, of continuous type, or discontinuous, or lymphatic endothelial cells, or whatever. So that's where you shift from medical imaging to microscope, like you are looking into.
And I think Serena Griffin has done all kinds of explorations of how to best extract that from Uberon. So I think I will set up another meeting with Chris and David, and maybe you can join as well to discuss this in detail. For now, we have a few more minutes left to have the last but not least demo of a sprint that just recently ended last week of the CCF exploration user interface that will become available with the first portal release for HubMap, and Bruce Hare is here with us, and he's going to present that. For those which need more background information on how this all works, there is another slide in this deck which lets you go to a poster that was recently presented at the All Hands meeting for HubMap. So feel free to check that out, including the CCF ontology, which some of you might be interested in. So I will give it over to Bruce. Thanks so much for demoing to us.
Thanks, Scotty. Let me share my screen. Can you hear me?
Yeah.
Okay. Okay, so I'll just jump right into it just so we end on time. This is the exploration user interface. So on the left, you have the partonomies, which are the ASCT tables that we're developing together inform and create this partonomy. I have kidney expanded because that's the one that we've put the most time into. But as these other organs mature, they also have, actually they already have some stuff in it too. So this is the partonomy. So this is the ontological side of the CCF. Then we have the spatial side. This is the reference body, and these are 4 reference organs that we have registered now. We are in development. We have many more reference organs in various stages of development. And the white pieces are tissues which have been registered via the registration user interface. Well, actually the spleen is a special scenario. They sort of hand wrote the registration, but in the next quarter of HubMap, we'll be introducing a registration user interface that applies to more than just the kidney. Right now, it just supports the kidney, but the framework is in place, as you can see, for other organs. And then on the right, you see all of the tissue samples from HubMap, which have been registered via the registration user interface. They're registered with the spatial side of the CCF. So you can see there are 43 samples. 28 donors. There's actually, it says 3 centers. There's 2. This is mainly from UFL, Florida, and Vanderbilt. The other one is the HubMap testing group, and that's just a testing sample that they have put in. So these tissues have been placed into the reference body, and you can zoom in. I have the tissue samples sitting out in front of the sample, in front of the organs right now. As Katia had mentioned, we had just finished a sprint, and we're starting our 4th sprint before that will be finishing up a lot of these features. So just for ease of communication, these actually fit inside of those reference organs, but I'm putting them in front just so it's easy to see. And right now you can click on one and it says what you clicked on. but that's associated with one of these samples here. Now, we have different samples that have been processed that are bulk samples, as in they ground them up and did stuff with that, but others which are spatially imaged and stuff. And so right now what you're seeing, this is a fake image. That's one of the things we're finishing up, the actual image viewer. We're integrating the test image viewer, which was also developed for HubMap. Actually, I can show you a quick. So it will be basically this, only in our tool. You'll be able to turn on and off layers, change their color coding. And that's what this is all about here. It's just, we're almost there, but we have one more sprint left to do that. So you can interact with the tissues, turn on different channels, color code them, layer them, have different overlays. And then the other thing here is like we can do query. You can click on the kidney and it would show all the samples which were associated with the kidney via the ontology. And we also have ways to filter, so you can filter by age, sex, and BMI. So you can see there are 18 samples that came from male and 24 samples from female. Let's see. And so we have a few different samples, a few different ways you can slice and dice the data. The cool thing you see here is you see these samples are registered very close to each other. So in a future release, we want to have the ability to say, okay, given a region on this reference body, find me all the tissue, all the cells that are associated with it, or all the tissues that have been placed in that same area, and we can start to do some very interesting queries with it. And nice thing again is we're all tied back to an ontology. So the other thing is we'll be segmenting these samples and extracting what cells are available in there, and those can be associated with our autonomy or the cell type ontology, and so that's kind of where we're at now, and we're looking to be good to go for the data release that'll be happening sometime this summer.
Thank you so much, Bruce. This was great. And you see some nice demo work comments in the chat also. So please check them out before you all go home. All right, everybody. Thanks so much. This was very, very informative yet again. There will be the next call in a month. So we do this monthly. So July 2nd, August 6th, you will see soon these invites in your calendar. If you have a few more moments and you want to talk with Bruce, please do so. I know some of you are very busy back-to-back Zoom schedules. I wanted to say thank you for everything you have done so far and come back. But if there are questions, please let Bruce know. I will stay on for a little bit. Thank you all.